All right, so today we're going to go over glycolysis. We might get into a little bit of the Krebs cycle. We'll see. But anyway, just so you guys know, for glycolysis, we're going to start off with glucose. Now, glucose is the main thing that is going to regenerate our ATP from ADP. And for each molecule of glucose, we will get approximately, your book says 32. We'll go through each step and do them individually and show you how many are generated. Okay. So for glycolysis, literally means sugar or glucose splitting. Glyco, glucose, lysis means to break or to split. So that's literally what glycolysis means, is sugar splitting. Okay. The nice thing about glycolysis, guys, is we do not need oxygen for this to occur. It can occur anaerobically. So whether you are a bacteria or a human, okay, you can go through glycolysis without oxygen. All right. So here is where glycolysis occurs. If you notice, I mean, here is our mitochondrial outer membrane right here. All right, so this is the outside of the mitochondria is where glycolysis is going to occur, and that is in our cytoplasm. So glycolysis occurs in the cytoplasm. Citric acid cycle or the Krebs cycle is going to be inside of our mitochondria, and there's an intermediate phase between glycolysis and the Krebs cycle called pyruvate oxidation. Very quick phase, very easy phase. But anyway, just to get into glycolysis, everything that happens in glycolysis, we are going to start with glucose. We are also going to start with NAD+. And some ADPs in there. Okay. We're also going to bring in, and this is an abbreviation for phosphate, it's PI. Okay. What we are going to do is the glucose is going to turn into pyruvate. The NAD pluses are going to turn into NADH. The ADPs are going to turn into ATPs. And we're also going to get some water, and you're going to think these are hydrogen. They are not hydrogens. These are pretty much just protons. Okay, we're going to get 2 H pluses or 2 protons out of it. All right. Now, this is all the transformations that are going to happen in glycolysis. Okay, what we put into it and what we get out of it. Now, there's going to be some different things I got to go over with you guys because this isn't the whole thing, but you'll see them in the diagram. Now, if we were to go into the book, you're going to see all these things, and oh my goodness, they look so complicated. I'm going to try to simplify it for us to the best of my ability, okay? So, let's start off. What do we start off with in glycolysis? Well, we start off with glucose. And I'm probably going to need 2 screens to do this whole thing. So, I will do half, I will erase, and then we will do the rest. Okay, so we're going to start off with glucose, and what we are going to do first is on our glucose. Wow, I can't write. Sorry, guys. On our glucose, we are going to attach a phosphate to it. Okay, so I'm going to draw a little circle here, and we're going to put our phosphate inside of that. Okay. Now for this, we got to get our phosphate from somewhere. Where does our phosphate come from? Well, we are going to take ATP and transform it into ADP. And in that transformation, guys, that phosphate, because we have 3 in ATP, we have 2 in ADP, that phosphate goes and attaches onto the glucose. Well, okay, that's good, but that really stinks because right now our ATP production is actually at a -1. We just use energy to split, or not to split, I'm sorry, to attach that phosphate to the glucose. Okay, so we're actually down energy right now. For this, any type of our reactions, guys, all right, we are going to need some type of enzyme in order to catalyze these reactions. You do not have to know all of these enzymes. I just want to show you that in this one, it's hexokinase. Anytime we have a kinase in here, it's going to be ATP and ADP. There's going to be some kind of transformation when we have a kinase. All right. From here, so we're done with the first step. And there's some other factors that need to come in here, guys, but I just wanted to show you the kinase, because anytime you see kinase, that's dealing with ATP and either taking it to ADP or taking ADP and putting it in ATP. But in reality, there's like other cofactors. And this one, it's a magnesium one, but we're not going to go into that. So after this, we are going to take our glucose and we are going to make it into fructose. All right. So if you remember, fructose is an isomer of glucose. And kind of cool here, the enzyme that does this is called isomerase. So glucose, fructose, and galactose all have the chemical formula C6H12O6. So when we do that, we use isomerase, that's our enzyme, and we still have our phosphate attached to it. Okay, let me get the lights back on here, guys. Very annoying, I apologize. All right, we're going green, saving energy, right? Okay, so after this one, guys, we are going to, once again, get more ATP in here. We are going to, once again, take another phosphate off of ATP, and now we're at ADP. Okay, so we still have that fructose, but in using another ATP and taking it down to ADP, we are going to put a phosphate on the other side of this. The issue with this is now our ATP is at a -2. We just use -2, or we just use 2 ATPs. So now, yeah, we just used 2 things of energy. So we're actually down 2 energies from where we started. Okay, and again, guys, ATP. Anytime you see ATP being transferred into ADP, our enzyme is going to have something to do with a kinase. So this one is phosphofructokinase. Okay, phosphate, yeah, we're adding a phosphate to the fructose, and it's a kinase. There you go. All right, now, after this part, guys, we are going to split our fructose, because how many carbons do we have in glucose? We have 6. This glucose, still 6. This fructose, still 6. This fructose, still 6 carbons, okay? What we are going to do is now, and I'm only going to do one of these pathways because they are identical, whether we're choosing side A or side B, those pathways are identical, okay? So, the next step is we are going to split our fructose into 2 3-carbon molecules, all right? On this side, we are going to have glyceraldehyde, and it's going to be attached to carbon 3, the phosphate. Okay, so glyceraldehyde 3-phosphate, and then on this side it would also be glycer ... Okay, aldehyde 3-phosphate. I didn't attach my phosphate yet, so let's do that. Okay. And yes, there is an enzyme that would do that. We don't need to worry about it, but it's fructose biphosphate, the enzyme. So we're not going to worry about that one, guys. I'm kind of toward the bottom of my screen here, so what I'm going to do is, if you want to take a screenshot of that, you can. But I'm going to erase all this, and then we're going to start back up at the top, continuing through glycolysis. All right, you know what? If you're taking a screenshot, we'll do glycolysis, part 1 up here, and then the second part I'll write part 2. So you guys, if you're taking multiple screenshots, you can stay organized, know which part is which. Okay. So again, I'm going to erase all this, and I'm only going to do one of these pathways. There's two. There's A, there's B. The same thing is happening on both. All right, so we're only going to do one of them. And just so you know, our ATP is still at -2. All right. So we're going to have our glyceraldehyde, 3-phosphate. Attach our phosphate to it. Okay. And this will be our part 2. All right. So we're going to take this and we're going to do some basic transformations here, nothing too crazy. And again, I know I keep repeating this, but we are going to do this twice. One on this side with our 3-carbon molecule and one on the other side with our 3-carbon molecule. So what we're going to do next is we are going to bring in some NAD+. And we are going to transform that. We're going to reduce it into NADH. Okay, so in reducing it, we get some NADH out of it. Now we get one on this side, we get one on the other side. Okay, we are also going to bring in some phosphate to this as well. Okay, so we're going to bring some phosphate in. How do we get the phosphate? Well, we are going to bring in phosphofructose kinase, and we're going to borrow a phosphate from that guy. So whatever this is, guys, I'm going to simplify everything. We're just going to say we have 3 carbons on it, our molecule here. Okay, so we got 3 carbons. Now, we still got our phosphate on that side, and on the other side, we are going to also attach a phosphate that we got from bringing in our phosphate there. Okay, so now we have 2 phosphates. All right. Next step. I know this seems kind of like we just did something and now we're going to do the opposite, but we are going to now start to take apart our molecule. So we're going to have 3 carbons, and on the 3rd carbon we're going to have a phosphate. Okay. We are going to remove a phosphate. And in removing this phosphate right here, we are going to take ADP, maybe one of those ADP molecules from before, and we are going to transform it into ATP. So now we got some energy, okay? But this happened twice. This happened on this side, this happened on the other side. So now, if you remember, our ATP up here was at -2. Now we're at 0, okay? Because we just gained 2, one from that side, one from the other side. All right. After this, we're going to do the same thing again. All right. And what we're going to do is there's a step in between, guys, where we're going to take the phosphate, and instead of on the carbon 3, we're going to move it over to the carbon 2 to make it more stable. I'm going to skip that step, okay? I'm just going to go down and we're going to take off that phosphate on the other side. We're going to get pyruvate out of it. In doing so, we are once again going to generate some ATP. One on this side, one on the other side. So overall, we get a +2. So how many ATPs did we form all throughout glycolysis? We formed 4. How many did we net gain? Only 2, because we had to use the ATPs from the beginning, okay, in order to attach those phosphates to our glucose. So yeah, we used 2, we made 4, so we only gained 2 in the end, okay? Also, in doing this, we're going to get some water out of that reaction, okay? All right, so that is the end of glycolysis, guys. All right, I tried to simplify it instead of giving you the very big structures of the book that they gave you. So you can screenshot that if you would like to. Okay. Okay. So products, just to go over our products, guys. I'll write those down real quick. Products. All right. We are going to get 2 NADHs, net gain of 2 ATPs. We're going to get 2 waters. We're also going to get 2 hydrogen protons. Okay? So those are our products of glycolysis. All right? Now, actually we could put one more on there. We also get 2 pyruvates, one on this side, one on the other side. Okay? So there's all your products, guys. All right. Moving on. So we're going to skip all this stuff here. Okay. All right. So now's going to be kind of like a fork in the road for us. We're going to start off with glycolysis no matter what organism we are using this for. So we're always starting with glycolysis. If we have oxygen present, we are going to begin to move over to the mitochondria. Okay. If we have oxygen present, guys, when we are moving over to the mitochondria, we can't just start the Krebs cycle, the citric acid cycle, just yet. We got to go through something called pyruvate oxidation first. Okay. So all that pyruvate oxidation is, guys, is we are going to have up here, that guy's our pyruvate. Okay. We are going to convert that into acetyl coenzyme A. That's that one there. Acetyl coenzyme A. All right. Now, here's the big thing with this. Pyruvate, it has 3 carbons. Acetyl coenzyme A has 2 carbons. Okay? So with this, we lost a carbon in this process, okay? Where did that carbon go? Well, that carbon, if we look at the diagram up here, that carbon escaped, combined it with oxygen, and now we have CO2. So what do we do with CO2? It's going to get released from our bodies. We're going to breathe it out, okay? Also in this process, we are going to take NAD plus and make it into NADH. Now, how many pyruvates do we have? We have 2 of them. So in reality, with pyruvate, okay, for each one pyruvate, we get 1 NADH out of it. Okay, we also get 1 CO2 released. Okay, but if we do our products of pyruvate oxidation, okay, we are going to get 2 acetyl coenzyme A's. We're going to get 2 NADHs. We're going to get 2 carbon dioxides from all that. Okay? So what we're going to do next is we are going to go over to the citric acid cycle. We're going to go over to the Krebs cycle. We're not going to worry about the NADH. We're not going to worry about the CO2 because we released that. We are going to worry about the acetyl, sorry, I forgot the L in there. The acetyl coenzyme A. That is what is going to go into our Krebs cycle. Okay? So when that goes into the Krebs cycle, guys, each turn of our Krebs cycle, the coenzyme A, each one is going to go through once. So in reality, we're going to take 2 turns, okay, 2 trips around the Krebs cycle, one for each acetyl coenzyme A. All right. Okay. So, sorry guys, let me get to where, okay. So just to give you a basic overview of the Krebs cycle, we are going to break down pyruvate. We're going to get some carbon dioxide out of it. So I know we just went through pyruvate oxidation and we breathed out some carbon dioxide that way. We're going to breathe even more carbon dioxide out from the Krebs cycle, get that as a byproduct. And we're also going to form the following. We're going to form 1 ATP, 3 NADHs, and 1 FADH2. Right? Now, with these guys, these are per turn. And we do 2 turns because we have 2 coenzyme A's. So in reality, our products of the Krebs cycle is going to be 2 ATP, 6 NADHs, and 2 FADH2s. Okay? So these are our products of the Krebs cycle. All right? And that is per our, if we want to go all the way back from glycolysis, per our 2 pyruvates being converted into 2 coenzyme A's. Okay, so now we are at the citric acid cycle. All right, if we wanted to talk about where this occurs at, this is going to occur in the mitochondrial matrix. All right, so that is going to be in the middle of our mitochondria, inside the inner membrane of the mitochondria. So we have our cristae, the finger-like projections going in to increase the surface area right there. Another one right there. Now in reality there'll be more than this, but it's going to be inside of those. So it's going to be all in this area right here. So that is going to be our mitochondrial matrix. So just to review, guys, we had glycolysis that occurred outside of our mitochondria in the cytoplasm. We had the Krebs cycle that is going to occur inside the mitochondria all the way in our mitochondrial matrix. All right. So here's our Krebs cycle, guys. I'm going to give you a shortened, easier version of it. Okay, so I will film that in the next video. Have a good day, guys.